Hey, Yueqing.
Hey, Aaron, how are you?
Good. How are you?
Good.
Good to see you.
Good to see you. Yeah, so everything's good. So the video, audio.
Yeah, it looks great.
All right.
Can you test out your slides?
Yes, I think should be fine. You can hear me. Yeah,
Yeah.
that's it. So, yeah, I heard a lot of people talking about you. So.
Can you try to share your slides just to see how they look?
Okay. Hold on. Let me share my screen. Is it good? Let me see.
No, I don't see your presentation.
Okay. Maybe wait. Is it because the delay? Yeah.
No, you're sharing your Zoom web browser.
Oh, okay. Let me see. Okay, then how about now?
Yeah, that's good. And then, yeah.
Good. Okay.
Perfect. Yeah.
Let me actually play one movie. Okay, I think it should be fine. You see movie?
Yeah.
Okay.
Yeah, very good.
Okay, very good. Should I stop sharing?
Sure.
Okay.
How's everything going?
Pretty good. Just came back from a GRC meeting in Italy.
Oh, nice.
Yeah, I heard people talk about it. That's why I said, yeah.
Nice.
Over there.
Yeah, I'm in Shanghai.
Oh, I was in Shanghai. Can you walk around?
I can walk around. Yeah.
Okay, that's good. I haven't been back for a while, so yeah.
Yeah. It's nice.
It's nice, good. Yeah, hopefully next year I'll be able to go back. Yeah.
Definitely by 2024. Maybe next year. Who knows?
Yeah, I have kids, so I want them. Yeah. To visit.
Right.
Yeah.
Right now there's a 10-day quarantine, so it's a little bit, and then it's also just hard to come back. But
yeah. Hi, it's Long.
Aaron, how are you doing?
Hey, how are you?
Very good. Nice to see
Yeah.
you.
Yeah. Haven't seen you very long time.
It's been a while.
Yeah. Last time I was still, I think, in Shanghai. See you.
Great. How are you doing in New York City?
Pretty good. Yeah. If you're talking about COVID, then pretty good. Yeah.
Yeah, it's already gone, right? It's everything. Yeah, we hopefully.
No, I think people talking about maybe it's rising, but it just less people care about it. So, but in terms of the case, maybe people worry about when it's coming, and we might see more cases. So yeah.
Hey, we get always.
Yeah. Have to. So yeah.
Right. You're working on sleep now, Yueqing.
Yes, I totally changed the field. No taste anymore. So yes.
Great.
But
it tastes already figured out. So no more secrets. Already separate.
Yeah. But I met someone at the GRC meeting. They actually very interesting insular codecs. So yeah, not necessarily taste related, but insular codecs. So yeah.
Hi, Ryohei. Good to see you.
Can you hear me?
Ryohei.
Yeah.
Nice to see you. Yeah.
Can you test your slides, Ryohei?
Yeah. Let me do that. Is it working?
Yes.
The full screen?
Great. Yeah.
Sounds good.
Thanks for coming.
Thank you for the invitation.
How long have you been at Columbia, Yueqing?
Almost 5 years, 4.5 years.
Okay.
But if you counting the postdoc, I've been here almost like 13 years, so very long.
Yeah, been there in 2009, right? It's time I
Yeah, 2009.
San
So very long. Yeah.
So you know Xiaoke?
I know. Yeah, we have
Yeah.
our lab. Yeah,
Okay.
definitely. I actually visited him this summer in his lab and his home. Yeah. I went to California.
Yueqing is also also IO alumni. So they're I guess you know each other in Shanghai, right? Or in
Yes, yeah, I know Xiaoke in Shanghai. Yeah. So yeah.
And what department are you in Columbia?
Now as pathology and cell biology.
Ah, got it. Okay.
Yeah. So I switched to another department. Yeah.
My friend Asa Abelievich was in pathology, I think.
Oh, what you study is neuroscience related?
Alzheimer's disease.
Oh, I see. Oh, interesting. Because now we are also interested in neurodegeneration, so maybe I can talk to.
Oh, and I've been trying for like 2.5 years to get Yamamoto to give a talk here. So maybe you can.
Okay, I know her very well.
Yeah, really good.
I can talk to her next time I see her.
Yeah, I've asked her like 10 times. She keeps saying no, but I think
I see.
eventually.
Yeah, she's busy. She's like one of the co-directors of the Neuro program at Columbia.
Yeah.
A lot of things. We are collaborating. I'm collaborating with her. That's why I'm saying neurodegeneration. So yeah.
Mm-hmm. Yeah, that would be great. And do you know Mimi Shirasu?
Yes, if so working flies, you study like circadian sleep in fly.
Yeah, she's also, we want to get her here.
Oh, yeah. But for her, we don't have any collaboration yet. I know her, but probably she's not very familiar about me. Yeah. But and then I know.
Yeah. The third guy is Alex Chavez. He's new. I'm still working on getting him. Yeah.
Maybe I don't know. Yeah.
Ryohei, how's your institute affected by the hurricane, or did you avoid it?
No, it was nothing. It was very quiet hurricane for us.
Okay, that's good.
Yeah. Seems like it went to the other side of Florida.
Okay.
Yeah.
So DeLong, did everyone at ION have to go in and watch like Xi Jinping's talk together and talk about it? Or could you...
Only I guess party members.
Oh, okay.
Which I'm not. I'm another another party, not a party. So yeah.
So watch a live stream or watch just the video? Watch live stream.
Some people watch on the WeChat, right?
Oh, I see.
A lot of you can watch on the cell phone if you want.
Yeah, I saw a bunch of people posting on WeChat, like sitting there or standing in attention, watching everything.
They're supposed to have a. Yeah, we get a slow started.
Sounds good. Okay. Hi, everyone. Another NeuroZoom week. Hope everyone's doing well. Before we get started with Yueqing and Ryohei, let me advertise next week's talk. So we have it's a UCSD edition of NeuroZoom, where we have Shu Chan and Christina Sigurdsson. So please come join us for those talks and let us know if you want to talk about your research. And now Ryohei can introduce the first speaker, Yueqing Peng.
Okay, Ryohei. Okay, it's my great pleasure to introduce another Ion alumni following last week, Yueqing Peng. So Yueqing has graduated from Nanjing University for his bachelor degree. After college, he went to ION, worked with Professor Aiko Guo with fly, so learning memory mechanism of fly. So after 2008, PhD, Yueqing went to work with Charles Zucker, initially in UCSD, and then follow Zucker to Charles Zucker to follow Charles to Columbia, where Yueqing did a series of amazing work to mapping out the sweet and bitter taste in bread using in vivo imaging. So after postdoc, Yueqing started his own lab in Columbia. So total switch gear to another field, which is neuromechanism underlying sleep. So today he will talk about it. I believe the first work come out from his own lab is a control of the non-REM sleep by medulla glutamatergic neuron. Okay, welcome with Yueqing. You might share a slide. Take it away.
All right. Let me share my screen. Okay.
Sure.
Can you all see the screen?
Sure. The current slideshow. Okay, great.
Okay, good. Okay, thanks for the introduction. Thanks for the invitation. Very glad to be here to talk about our new work on sleep. So my title is Control of Non-REM Sleep by Medulla glutamatergic neuron. So first, allow me very briefly introduce our lab. So we study neurobiology of sleep in both normal and diseased brain. So we are mainly interested in 3 areas. One is the neuromechanism that controls sleep. So this will be our topic today. And the second is the memory function of sleep. So we are particularly interested in neuroactivity during sleep that might contribute to memory consolidation. And the third one is in collaboration with other labs at Columbia University. We're also interested in understanding the relationship between sleep and brain diseases. Right now, with a focus on epilepsy. So this is our lab interest. So first about sleep. I think everyone here knows that there are 2 types of sleep, REM and non-REM sleep in mammals. And of course, mice also have both REM and non-REM sleep, and it shows quite a similar EEG pattern. I think for today's talk, I will mostly focus on non-REM sleep. So we are interested in the neural circuits that control sleep, and decades of study has revealed essential roles of neuromodular systems in regulating wakefulness. So these are brain structure located in mostly in brainstem and hypothalamus. But early work also identified a group of GABAergic neurons in ventral lateral preoptic area, called VLPO, that are involved in promoting non-REM sleep. So this is most famous, I call the sleep center. But recent work has identified several other brain areas that are involved in non-REM control. So here's an example I showed here. Notably, a lot of works are from Young Ta's group in UC Berkeley. And we also interested in to identify new brain areas that can control sleep behavior, particularly in sleep initiation. So sleep initiation is you can see it as a process, a transition process from wakefulness to sleep. On the normal physiological condition, always transitions to non-REM sleep. So basically, is the transition from wakefulness to non-REM sleep. We consider here as a sleep initiation process. And so among all these non-REM related region, the VLPO, as I mentioned, is the most studied region. So like here, for example, very earlier that C4 study shows there's a correlation between the number of C4 positive cells in VLPO and the amount of non-REM sleep. As you can see, this linear correlation here. Then consistently, electrophysiology recording shows that VLPO sleep active neuron shows the firing rate of this neuron correlate very well with the depths of sleep states, non-REM sleep states, usually indicated by the delta power. As you can show here on the left is the delta power, and on the right here is the firing rate. But interestingly, if you look at carefully, so here this VLPO sleep active neuron, their firing rate actually quite low in the transition period. If you compare that to the actual sleep states, and their firing rate actually increased further from light sleep to deep sleep. So for me, this finding suggests that maybe VLPO neuron might be involved in sleep maintenance. The other neuron will be involved in sleep initiation. So we think that neuron projecting to the VLPO might be involved in sleep initiation. So to help identify this neuron, we performed retrograde labelings and by injecting retrograde coronavirus to the VLPO in a report line, AR9 report line, which is a flux TD tomato report line. So then we use brain clearing and whole brain imaging, try to identify those input neurons to the VLPO. So here is an example of whole brain imaging. So as you can see, the very bright spot is the injection site in VLPO. And I think from now on, I will try to use pre-optic area, POA, instead of VLPO, because we use a lot of viral injection. Often it's not very restricted to the VLPO side, just the POA. And you can see that most labeled cells are located in forebrain, but there's also a group of neurons in the brainstem. This will be our most, we're particularly interested in neurons in brainstem, because early transition studies suggest the existence of sleep promoting centers in the brainstem. So here we found a group of neurons, if you can see here, in the ventral lateral side of the medulla. And so here, if we do maximum Z projection on either coronal section here or a horizontal section, this is like a Z projection. These are cells here, and we also can do a 3D reconstruction of these labeled cells in the brainstem. So this is their locations. And so then next, we try to figure out what type of cells they are in both this ventral lateral medulla, I call it VLM, or in this preoptic area, or POA. So first in the VLM, and we perform the co-staining of retrograde labeling and use a glutamatergic or GABAergic neuronal marker here. And we show that most retrograde labeled cells here is a TD tomato positive cell, are actually glutamatergic cells and not GABAergic cell. And then we also want to know in the POA region, which cell type actually receive input. Because as I mentioned, the earlier work showed that the sleep active cell in POA are GABAergic. So we want to know where the VLM neuron actually synapse to GABAergic neuron in POA region. So to do that, we use this rabies monosynaptic tracing. So in a GATA2 animal, we inject rabies virus in the POA region, and then look at the input neurons in other brain area. So indeed, we found that POA GABAergic neuron receive input from VLM. If you can see this green label cell is their input neurons. So from these two tracing study, we show that it suggests that VLM glutamatergic neuron are connected to POA GABAergic neuron. To actually demonstrate their function connectivity between these two regions, we then perform the slice recording. So we try to patch GABAergic neurons in POA region while optogenetically stimulating the channel rhabdosis expressing VLM axon terminals of VLM glutamatergic neurons. So to do that, we inject this create dependent channel rhabdosin in VOM, then inject this MDLX M-rubies in PLA. So here, this MDLX promote allow us to target GABAergic neurons in the PLA. So then we patched, say, this M-ruby positive cell is supposed to be a GABAergic cell. And here's an example. As you can see, channel rhabdosin activation can induce EPSP, and which is blocked by glutamate receptor antagonist here. This is combined the mix of both AMPA and NMDA receptor. And this effect can recover after washout. Here is a washout. And then here's just a quantification, and so shows this actually VLM glutamatergic neuron are connected to PLA GABAergic neuron, and they release glutamate. Okay, next we want to look at the activity during the transition period in vivo. So, and first thing we did is we tried to use CFOS activity experiment. And so here we have 3 groups of animal. One is motor control, and the other is sleep deprivation, or SD here. The third one is recovered sleep, or ICE. So to sleep deprived animal, we designed intermittent a device that we can do intermittent swiping movement. So just basically it can move a rod back and forth in the cage, and then we can stop for a while and move again. So here we can show, because this intermittent pattern, it will force mice to temporarily wake up when they are sleeping. So then we can create a lot of awake to sleep transition. So here is an example. If you there's a lot of like sleep, wake, sleep, wake, sleep, wake. So we hope to create a lot of transition to induce to maximize CFOS expression involved in this process. And this recovery sleep group, so we waited 2 hours after sleep deprivation, and then second animal. This 2-hour period allow animals to actually fall in sleep, have a lot of non-REM sleep, but they stay in sleep state. There's a less transition actually. So we did some quantification here. This SD mice has the most transition time. And then we look at CFOS in the VLM region, and we found in SD mice, we found significantly more CFOS positive cells. And then by co-staining, we found this CFOS positive cell are also glutamatergic cells, not GABAergic. So these suggest that VLM glutamatergic cells are involved in wake to non-REM transition. But of course, we all know that CFOS has very limited temporal resolution. And to further study the activities of this neuron in the wake and sleep cycles, we then perform in vivo recordings by using microendoscopic calcium imaging in the brainstem. So here is one imaging station showing that the top is the brain state. We color code the brain state. Here, orange is non-REM sleep. Gray is awake. Purple is REM sleep. Then next is EEG spectrogram and EMG. Then a few examples of calcium traces. And among them, so for example, here this number 6 cell, we consider shows a lot of activities during non-REM sleep. We call this non-REM active cells. And so then we try to quantify the activities of these cells in different states. We used an index called the Selectivity Index. So basically, it measures cells activity, which side they prefer. So for example, here NW is non-REM versus wake index. If it's positive, that means the cell shows high activity during non-REM sleep, low during wake period. So same thing is here for the y-axis. For the RW is REM versus wake. If it's high, then If it's positive, that means the cell shows high activity during REM. If it's negative, means showing high activity during wake. So in this way, we can know that for each cell, what is their firing profile. And for each dot is one cell. So this blue labeled cell we can see the non-REM active cell. So if we quantify the activity here, you see they show highest activities during non-REM sleep compared to wake and REM sleep period. Then next thing we did is we analyzed the activity during the transition period. So interestingly, if you can see here, this non-REM active cell actually showed increased activities from wakefulness to non-REM sleep transition, but the activity almost remained unchanged during the transition from non-REM sleep to wake. So it seems like the activity is quite specific, to the wake to non-REM sleep transition, suggest their functions in this transition. Okay, next we try to know their function in vivo. So we try to see whether they are required or sufficient to promote transition. For that, we want to do some manipulation. So first, we use chemogenetic inhibition to specifically target this pathway. We inject a retrograde crease in POA, then create-dependent inhibitory drug in VOM, and then just inject CNO or settings to look at the effects on sleep. And we found that chemogenetic inhibition actually increase wake time and decrease both non-REM and REM sleep time, which is consistent with our expectations, because if these cells are required for the transition, inhibition will suppress sleep. And so as we see here. And then we did a fold analysis to see what the decreased sleep, decreased either REM sleep or non-REM sleep, actually is caused by the decreased the bouts. We call it bouts number, not duration. The bouts consider like episodes, how many like small episodes they have. Now we see with inhibition, there's less bouts, suggesting the less transitions from wake to sleep. So yes, try to highlight here. So next we did chemogenetic activation, use a similar two-viral strategy to target a POA projecting VOM neuron. And we see opposite. We see that activation can increase non-REM sleep, and this increase mostly come from the bouts, also the number of bouts, suggesting more transitions happen. And the lastly, we used optogenetics. The reason is that, as we know, that the transition from wake to sleep happens very quickly, usually in a time scale of seconds. Chemogenetic manipulation don't have this time resolution. So to look at each trials, we turn to optogenetics. And we use the two strategies. One is use retrograde labeling. So we similar to the strategy we used for the chemogenetic manipulation. So we use two virus, retrograde Cree and then a Cree-dependent channel opposite in the VOM. And we found that optogenetic stimulation also can trigger the transition from wake to non-REM sleep. And another we grew two animal direct inject to the VOM and then activate terminals in POA. Similarly, we can induce non-REM sleep in a wake animal. So as you can see here. And then I should say that the VOM neuron, other than projecting to the POA, they also projecting to other brain regions that are not involved in sleep. For example, here is a region called ZI, in theta, and the other is a P3 nuclear or the PAG area. So we are now looking into this pathway in sleep behavior. Hopefully we have some data later. With that, so to summarize, I hope I showed that VLM glutamatergic neuron project to PLA GABAergic neuron, and VLM glutamate neurons showed increased activity during the transition from wakefulness to non-REM sleep, and then chemogenetic silencing of these neurons suppress non-REM sleep, while activation actually promote non-REM sleep. And finally, optogenetic stimulation of VLM to PL pathway can promote non-REM sleep in wake animal. With that, I want to thank my lab. Most of the work are done by Sasa Tan here, and also my collaborator and findings. And also last one, very important, we are looking for a postdoc. If anyone interested in sleep, please contact me. Thank you very much. I'm happy to take any questions.
Great, thank you, Yueqing. A good talk. Now we'll open for questions. Yeah, I'm particularly curious about the one piece of data is in activation of the secretary can induce the awakeness to the non-REM sleep.
Yeah,
those kind of sleep initiation something like that.
Yeah, that's why we tried. So the purpose is try to say that this neuron involving initiation, although probably need more evidence to show that. I think yes, this is exactly the purpose. So that's why we separate this trial. So we look at specifically in awake animal, what is the probabilities if we activate can trigger into sleep. So here it's like it's not 100% yet. It's like 50% of the chance we can induce sleep. Yeah, if we activate this pathway specific activation. Yeah.
Okay, Kevin, please. Okay, unmute yourself.
Hi, Yueqing. Thanks for the talk. It's very interesting. So, in continuous of those questions, so for initiation, is there any specific requirements for the activation level of or activation pattern of these VLN neurons to initiate sleep? Because you mentioned that it's about like 50%.
Yeah. So yeah, we are
successful rate in initiation, right?
Yeah, we are very interested in the firing pattern. So our imaging data shows that indeed they increase activities in the early period of non-REM sleep, but because I think due to the resolution of classical imaging, we cannot tell very precisely the firing patterns. We need to do some I think in vivo electrophysiology recordings to figure out the patterns, especially the very short period. But actually, in terms of this probability, because of the time, I don't want to go too complicated. So here I will show that if we activate these retrograde label cells or terminals, is like 50%. But we also actually try directly activate the somas in VLM. It has much higher successful rate to induce non-REM sleep. But there's other. So if I have time to show, I think here. So here is a much higher probability. It's almost 80% if you activate the soma directly. But the caveat here is that it happens when we turn off the light, not when we turn on the light. And also we notice that if we activate during sleep, it actually can wake up animal. So I think because this here is like non-specific activation, there are like sleep active neurons in the region, but they also have other neurons, especially the motor-related neuron in the region. So, but the very interesting thing here is if we look at the probability of to induce this transition, here has a much higher probability. So make us wonder that maybe there are some VM neurons that project to other area, not project to PLA, also contribute to the initiation. So we try to look at it. That's why I say we look at other downstream pathways. Yeah.
I see. And another question is the medulla actually is like a key mediators of the motor controls. So, and also it's been like involved in like PD development and etc. So, is there any problems in the PD patients or PD animal models that the VLM area there's like a neurodegeneration or any abnormal neurophysiologies?
That's very interesting point. So I haven't seen the paper yet, although as I mentioned, this region, there's a group, they published this neuron could involve in control locomotions. But whether the same neuron could be involved is so far not sure yet. Yeah, we are actually interested in Huntington's disease, also a motor neuron related disease. So probably we'll look at this in the motor function later. But so far, right now, I haven't seen that yet. In my model, this motor-related neuron should be separate from the sleep active neuron, although they are intermingled, maybe a little bit overlap in the region. Yeah.
Okay, great. Thank you. Thank you very much.
Thank you.
Okay. Any more questions? Okay. Do you want to ask questions yourself in the chat board? Ying?
Yeah, I can read. What is the ratio of ratio labeled, for instance, positive neurons of the glutamatergic neuron in VLM? That's to say is a good marker. So the ratio is what I showed is like close to 80%, and also showed like almost 0 for the GABA. That means there's like 20 something remaining. So we don't know what are those 20 cells yet. We try some mark, but not very successful yet. So, but for the glutamatergic marker, it's close to 80%. Yeah.
Okay. I have one last question about the previous slide you showed. You're activating the POA projecting VOM soma. Then you find that increasing REM sleep at some point, right?
Increase non-REM sleep, REM sleep?
Yeah. You showed the previous slide. You showed you're activating the circuitry specific activation.
So,
you showed.
No, so here is also non-REM sleep. Not REM is very little. RAM is a purple.
Oh,
so
okay,
I probably use different colors. Yeah, non-RAM. Sorry, I have this RAM word here underneath this curve. Sorry, it's misleading. Yeah, it's non-RAM, not RAM, not RAM. Yeah, not RAM.
Okay.
Also, I would say this region is very close to another group of non-RAM promoting neuron that is discovered by also Yang Da's lab, but those are GABAergic neuron.
You mean VLM, right?
They call it LPGI. I think it's close to that. Not exactly same, but very close. Yeah, but they are GABAergic neuron. Yeah,
this is not right next to the VLM.
Yeah, we think maybe there are some local connections between those. Yeah.
Okay. Cool. Great. Thanks, Yueqing, for
Thank you.
talk.
All right. Thank you.
So, Aaron, to introduce Rohan.
Great. Thanks, Yueqing. Awesome talk.
Yeah.
It's now a great pleasure to introduce Dr. Ryohei Yasuda as our next speaker. Dr. Yasuda did his undergraduate and PhD training in Japan. He received a PhD in physics in Yokohama at Keio University. He, as part of his PhD thesis, he used single molecule measurements to study the mechanism of a subunit of ATP synthase and discovered it functions as a rotary motor. He then came to New York to do a postdoctoral fellowship with Karel Svoboda at Cold Spring Harbor Laboratory. And here he developed a method to sensitively quantify protein interactions, calcium concentrations using high resolution microscopy within neurons, but not just within neurons, subcellular compartments within neurons. He, throughout his career, he has pioneered sensors for key signaling pathways in the cell. These, for example, reporting on the activation status of small G proteins like RAS and RAC, not just reporting them within the cell, but in specific parts of the cell or in specific dendrites or within dendritic spines. He started his own lab at Duke, but then moved to become the scientific director of the Max Planck Florida Institute. And he's been there since 2012. He's won many awards, including the NIH Director's Pioneer Award. And he is a wonderful colleague and a dynamic scientist who has made many important contributions, not just in tool development, but into insights about signaling pathways within neurons. So we're delighted to hear the latest, Ryohei.
Thank you very much for the introduction. So let me share my slide. So, okay, good. So we have been working on intracellular signaling because we believe that intracellular signaling is involved to some degree, at least, to long time scale of the brain activity. Basically, we think that neural activity and intracellular signaling has some feedback loop, eventually regulates the behavior and neural circuit at time scale of seconds to days, or maybe years. And this longer activity is required for our learning memory and adaptation and many brain activity in the long time scale. So we believe that intracellular signaling plays an important role in basically connecting the different time scale. So we have been working on understanding intracellular signaling in neurons, mainly in pyramidal neurons and dendritic spines, because we think that dendritic spines, of course, is important structure for learning memory. And also at the same time, it has morphologically very interesting features, like it has microdomain and compartmentalized to some degree to support independent signaling to increase, basically maximize the information storage. But at the same time, of course, it's a part of a cell, and therefore they need to have communication between different compartments, micro compartments, and eventually to the nucleus. Now, when we're talking about intracellular signaling, it actually is a very complicated system because of the number of species of the proteins expressed in the neuron and their extensive interaction. So this signaling system, basically, it's made of 1,000 molecules and interacting with each other, and they form a kind of activity network. And this network somehow to basically connect, for example, neural activity, right? Receiving some input from outside world, and then do some information process, and eventually it will give outputs that might be some kind of synaptic plasticity, for example, LTP or LTD. In our lab, we extensively use single spine LTP because it's convenient to understand signal transduction in vitro. This is in brain slices, organotypic slices we use. And to understand signal transduction, it is probably better to have some kind, especially and temporally regulated way to simulate, because otherwise you really don't know actually where calcium comes from, where what kind of stimulation you are having to trigger activation of intracellular signaling. So this glutamate uncaging, it's convenient. It produces structural plasticity and functional plasticity in single spine, not in adjacent spines. And by using this system, basically we can reliably induce plasticity in one spine and at the same time observe signal transduction to understand what kind of biochemical reaction occurring in single spine. Okay. So in our lab, we are focusing on two aspects of studying intracellular signaling and like monitoring activity and manipulated activity of the proteins. And these two are basically required understand and interrogate the signaling system in single spines. Today, I'm going to talk about pretty much about monitoring activity. Manipulated activity, photoactivation of signal transduction is also another interesting area to connect the behavior and intracellular signaling and also different components with intracellular signaling. But probably if I have a different chance, maybe I can talk about it. So to image intracellular signaling, we have been using a system called two-photon fluorescence lifetime imaging microscopy. This is a combination of two-photon microscope and lifetime imaging, fluorescence lifetime imaging. And this is a very robust and quantitative way to measure fluorescence resonance energy transfer or FRET. Basically, we use a short laser pulse to excite fluorophore and measure the decay occurs in nanosecond time scale. And basically, this lifetime decay slope is sensitive to the fluorophore distance, and therefore we can basically calculate how much proteins are bound to each other, which is useful to basically convert signaling events into fluorescence lifetime signal. So, of course, we need a biosensor to measure intracellular signaling, and that's actually most of our efforts come from. And we try to scale this up, but still there are a lot of obstacles just to create single sensor and validate. So the throughput is pretty low, but we created about 20 sensors so far. Many of them works for imaging single spines. And then we get a lot of different aspects of signal transduction, like spatially, how the signal transduction spatially do you see the arrow by the way?
Yep, we can see it.
Yeah. Oh, okay, good. Then I don't need that. Yeah. Okay. So basically, we can understand how signal transduction is compartmentalized in response to spatially restricted stimulation and spatially restricted plasticity. So basically, we see some signal transduction stays in the spine and eventually spreads to adjacent spines and basically influence the plasticity in other spines. And something else spreads to the entire dendritic branch, and sometimes we see actually activity goes all the way into the nucleus. So this kind of spatial computation, I would say, is important for understanding actually what each molecule is doing. And also, of course, as our motivation indicates, basically this temporal aspect is very important. Like calcium comes into the spine. Calcium signal is very short in the spine. It lasts only for 100 milliseconds, and that is relayed by several calcium-sensitive kinases like CAMK2 or PKC, which lasts for several seconds and further relayed by different proteins in the spine. Okay, so first I'm going to talk about PKC, protein kinase C. The study is pretty much done by our postdoc Leslie Colgan. And the motivation is that PKC, of course, is known to be a very important molecule, like calcium sensitive and so on and so forth. But actually, it's not so well studied because we don't have really great tool to activate, inhibit specific isozymes, and also to read out the activity of different isozymes. Right? So basically, Leslie tried to to image the activity of individual PKC isozymes. So she came up with two ideas. One is to measure the trafficking of PKC associated with activation to the membrane. So she called it iTrack, isozyme-specific translocation of C kinase. Basically, she fused PKC isozyme with the GFP and membrane with RFP. And basically, translocation causes a FRET, which we can read with fluorescence lifetime. Another one is iDocs, basically looking at the binding between GFP fused isozyme and RFP fused to substrate. So both of them, as you can see, we use full-length PKC, and hopefully that whole full regulation occurs in these fusion proteins. And so she made sensors for all these different PKC isozymes. And for example, classic PKC, which is calcium sensitive, shows activity in response to glutamate uncaging. So this is one example of activity of PKC alpha. And basically in the spine, the kinase is activated in response to every uncaging pulse. This is a real-time movie. So you can see that actually when we stimulate the spine, we observe activation of PKC alpha and decays within about 0.6 seconds. There's no accumulation or integration by this kinase, and therefore we see kind of activation goes up and down, up and down. This is actually y-axis is activity, and y-axis is time scale, time in seconds. Sorry that I forgot to put axis here. This is a spatial pattern. We have PKC activity high in the spine and decays in a couple of microns. So again, this is a spatial, temporal pattern of PKC activity. It's very fast. Okay, so that's about PKC alpha. Actually, it has interesting interaction, interesting regulation by BDNF, which is released from the spine, and which activates track B, and actually which is required for PKC alpha activation. So it's actually there's a reverse direction of activity from the longer time scale to short time scale. Anyway, it's a kind of interesting integration point. I talk about the kind of unpublished study about PKC, novel PKC. So novel PKC is not calcium sensitive. It's actually activated only by deacylglycerol or DAG. So basically, Leslie wanted to know if novel PKC is required. After several experiments, she came up she found that PKC delta is important for structural plasticity and LTP. And for this, actually, she developed a tool to specifically inhibit PKC delta isoform by mutating PKC delta ATP binding pocket, which fits to this small ATP analog drug 1-NPP1, and therefore we can inhibit only this mutated PKC and remaining all other PKC intact. And so in the knockout background, she overexpressed this protein and rescued the plasticity. Okay, so knockout, okay. I don't have the knockout data here, but actually it just rescues the plasticity. So in the vehicle, you can see that structural plasticity occurs. We have kind of characteristic pronounced initial phase followed by sustained phase. So all phases are intact. Now we can basically let's see. We can inhibit the PKC delta and here the blue one from the beginning, and then we don't have plasticity. Okay. If we add the 1-NPP1 to inhibit PKC delta after the induction, which is indicated in black, we don't see any inhibition. Okay. So PKC delta is required for induction, not for maintenance. So she, of course, measured the activity of PKC delta. So the PKC delta is not calcium sensitive, and therefore it requires kind of secondary signal that makes the kinase much slower than PKC alpha. So this is activation profile, time scale, so lifetime change. So this activation causes the decrease of PKC over the lifetime, and you can see that it's activated and it comes back. There's some sustained activity actually remains for a very long time. Actually, we never have seen the decay, so it seems like actually it can activate for a long time. And actually, it's also there's some spreading of activity into the dendrite, indicated in red. Okay. So some pharmacology we did. So basically, DAG it's activated by DAG again, and DAG is produced by track B, actually, which activate PLC. So when calcium comes in, BDNF release, activate track B, and activate PKC delta. So we basically found that this pathway activate PKC delta from pharmacology and genetic deletion BDNF. Okay, now let's wonder actually what this long-term component and long kind of spreading component of PKC delta. And she decided to work on the connection, activation spreading of PKC delta, and she thought it might spread all the way into the nucleus. And previously in 2013, a long time ago, we found that actually single spine, 3 to 7 spine activation causes activation of ARC in the nucleus. And actually, it PKC dependent when we activate. So this is a nuclear ARC activation in response to 7 spine stimulation, and 7 spines not simultaneously activated. It's very slow protocol. We stimulate one by one, and then we observe activation in the nucleus. The red one is actually with the PKC inhibitor BIM. It's non-specific PKC inhibitor, but basically we observe the decay. So and PKC delta is slow kinase and also spreading, so it might have some role in this kind of activity. So let's decided to measure the activity of PKC delta in the primary dendrite while stimulating 5 different spines. Okay, again, we stimulate 5 different spines one by one, very slowly. There's no electric coupling between this stimulation, basically. And then looked at the activity in the primary dendrite, and we observed actually pretty significant activity after 5 spine stimulation. 1 spine seems to be not enough to activate, but 5 spines we observe very significant activity. So there is a activation occurs and propagate all very long distance to the nucleus. To the primary dendrite. We also looked at the Krebs activity. We actually happened to develop Krebs sensor several years ago, a few years ago, and we basically found that so activity of Krebs in the nucleus activated in response to 5 spine stimulation, and which was blocked by specific PKC delta inhibition, okay, which we introduced a bit time ago. Okay, so that's the story about PKC delta. I think, okay. So basically, PKC delta is activated locally to induce LTP, and at the same time, it actually have long distance signal activating Krebs in the nucleus. I think maybe I use 5 minutes trying to give you some introduction about insulin growth factor anyway, so this story is done by Mori too. Basically, insulin like growth factor is one of the family of insulin peptide. Insulin IGF-1 and IGF-2 have very similar structure, and IGF-1 and 2 can activate IGF-1 receptor. Insulin activates insulin receptor, act as growth hormone, and there are some kind of crosstalk between these 3 signals. They are synthesized mainly in the liver and across the blood-brain barrier, but actually recent studies suggest actually a lot of neurons actually, and also glia can synthesize this IGF-1 and the IGF-1 and 2 signaling seems to be implicated in a lot of different diseases and mental disease and so forth. And it's not including learning memory. So I think it is kind of important molecule. So it's a tyrosine kinase. It's activated by binding IGF-1 and recruit downstream molecule like PI3 kinase and activate gene transcription and so forth. So first, our surprise that Mori found that actually IGF-1 is very specifically expressed in C1 pyramidal neurons. Not much in CS3. And this is really striking. Actually, when we sparsely deleted IGF-1 from, some neurons, like these green neurons, knockout neurons using a frogs animal and sparsely infected with GFP, flex GFP and CAMK2-3. And we actually could remove this signal, suggesting that basically these neurons actually synthesize IGF-1. And interestingly, when we remove IGF-1 gene sparsely from the neuron, actually we also observe, also observe attenuation of structure plasticity. Those are time course of structure plasticity. Kind of reduction in initial phase, actually a lot of reduction in the sustained phase. And so in knockout neurons, so knockout neuron is red and wild type is black. We can rescue it by adding IGF-1 is indicated in blue, partially rescued by just birth application of IGF-1. And also electrophysiological RTP seems to require IGF-1 synthesis in the postsynaptic neuron. So, so this is wild type is black and red is control. I skip this. So we developed sensor for IGF-1 signaling by basically modifying IGF-1 receptor, attaching GFP, and recruiting molecule with RFP, and measure the threat between these molecules. We can measure IGF-1 concentration in slices, brain slice. Sorry, it's not acute slice; it's an organotypic slice. But we see the signal in basically dose-dependent manner. So, and maybe you can predict actually where the story goes. Basically, when we stimulate single spine, we observe the activation of IGF-1 receptor. So suggesting that IGF-1 probably is released from the spine and activating the receptor on the same spine. This activation requires, this activation in x-axis and y-axis time, and gray is the activation. It requires glutamate receptors, NMDA type glutamate receptors to activate. When we delete the gene of IGF-1 from these neurons, we observe the attenuation of the signal. So this suggests that IGF-1 is synthesized in neuron and release IGF-1 from the spine and activate IGF-1 receptor on the same spine. So that's basically the story. We believe that calcium actually causes IGF-1, and since IGF-1 is actually synthesized, and that calcium influx triggers the release, which comes back to the same spine, activates IGF-1 receptor, and causes spine plasticity. So I think that's pretty much it. I think I have some story, but I think I probably skip and just thank my colleague who actually did all these experiments. So Mori too is a grad student, a very talented graduate student, went to UC Berkeley as a postdoc now. And first story about PKC was done by Leslie Colgan, and a lot of help from other members of the lab, like Anand, who did the EFIS, Paula also helped electrophysiology, transgenic and viral injection, transposed by Yueqing, and actually gene manipulation by Kathy. And thank you for your attention.
Thanks, Ryohei. Awesome talk. And now we're open for questions. John?
Hey, thanks. Hey, Ryohei, great talk. I have a question about the IGF story.
Yeah.
As I'm sure you know, there are quite a few IGF binding proteins in the encoded in the genome. And do you have any thoughts about or evidence for how they might sculpt what's going on here in the system?
Yeah, I think this is a very interesting question, and we are actually actively working on IGF binding protein now, because I think they have essential role in basically capturing IGF-1 and maybe even for storage. So we don't have any evidence right now. Any results actually how these proteins may play a role in the storage or regulation of IGF-1, but we are very curious actually and to see actually how IGF-1 is regulated. Great question.
Great, thanks a lot.
So Ryohei, I was intrigued by the finding of signaling from the spines to Krebs in the nucleus. And I'm just wondering, what are your thoughts on how that is sensed? Or does it matter where the spines are? Does it matter how many there are? Are the ones closer to the nucleus? Or...
Right. So we have done these experiments kind of in the first publication, and so we basically use arc sensor instead of PKC delta. But yes, so it actually, if you stimulate, for example, the distal dendrite, it takes more time for the from spine to the nucleus. It's basically a very interesting time course. We see very flat signal for some time, and after 30-40 minutes, it start to increase. So for the distal dendrite, the answer is that it takes more time, but it can still activate the nucleus. And the spatial pattern, actually, you need to stimulate more than 2 dendrites, two dendritic branches to activate. If we cluster all the activation, all the LTP in one dendrite, actually, we don't see any activation of the crab. So we need to have at least 2 branches.
Okay, got it. Next question from Yuki Kitanishi.
Thank you very much. Can you hear me?
Yes.
Yes.
Yes. I'm also interested in the same point.
It seems like.
Hey, Yuki, it looks like you froze. Maybe come back for the question when you can. Mingjia.
Hi, Ryohei, great talk. I just have a quick question on the PKC part. So when you were manipulating PKCs, the relationships of the PKM and the different forms of PKC, how would you be able to specifically monitor the role function of PKM?
Yeah, that's something we are trying to. Actually, we are trying to also trying to do. We haven't done yet. I can't tell you the results yet because I don't know yet. we are trying to do it. Yes.
Oh, okay, yeah. Maybe we can sometime talk offline.
Sure. Yeah, sounds good.
Yeah. Thank you.
Hey, Yuki, are you back?
So can you hear me now?
Yes.
Yes.
Oh, sorry about the problem. So I'm also interested in the same thing, and especially the difference between the MAP kinase ERK and PKC delta. So first of all, I found it so interesting that the activity maintenance after activity. So you've showed us that even after activity, there's kind of sustaining activity inside the spine about the PKC delta. So first of all, what is a mechanism that make it activating for about whole time after activity?
Yeah, that's a great question. And so we are speculating that there's a probably positive feedback because to enhance signal, it requires some amplification to have signal from the single spine to the primary dendrite because the volume is very different. So we think that some amplification mechanism is necessary, and speculating that maybe it's caused by positive feedback loop between PKC delta and other kinase. We haven't found yet actually which is downstream and which is actually coming back to PKC delta yet. But just that's I think it's a likely scenario, I think.
Thank you very much. And also I found it so interesting that PKC activating PKC delta activating several spines integrate into the nucleus and after all to the crab activity.
Yeah.
And also you've shown in the previous report, you've shown that MAP ERK activity soon goes into the nucleus. So is there any difference about the spreading speed of the activity between PKC delta and ERK? And is a crab differentiating between ERK activity and PKC delta activity?
Yeah, I think the activity seems to be similar. And actually, but so we actually measured ARC activity while inhibiting PKC delta, and we couldn't find the link between these two proteins. But the spreading speed is same, and both targets to crabs. So I don't know actually how they act. Probably they need to do in parallel and diffusing its diffusion speed also seems to be similar. So still, it's a kind of mystery how they interact with each other.
Okay, thank you very much.
Okay, Nathan.
Oh, hey, Nathan.
Hey, Ryohei. How you doing?
Pretty good.
Good. Great talk. I know that you typically haven't focused on this kind of thing in the past, but is there any reason to believe that in these multiple dendrite stimulation types of settings that there's an addressing scheme that's maintained for the stimulus sites? So for instance, if you restricted your 7 spine stimulation to 2 dendrites, is there any reason to believe, for instance, that some of the dendrite targeted mRNA might target specifically to those dendrites, or do you think this is more of a global signal?
That's actually an interesting point. So yeah, I think actually when you have some cluster activity, it seems like so it doesn't. Well, I wouldn't say it doesn't get into the nucleus. Actually, if you have also some signal comes to other dendrites, yeah, they can be integrated, right? So, but if that's a stimulation alone, I mean, if you have just all inputs in one dendrite, yes, it is restricted. And actually, that dendritic translation, local translation, seems to be kind of an interesting target for this event. And we actually found that each dendritic segment is relatively quickly saturated. So that actually is the reason probably that actually you don't have more integration by adding spines in one dendritic branch. And that probably actually causes local computation. And when you add more signal, then two saturation causes, of course, some additive effects to cause further activation in the nucleus. So I think that's the mechanism. And actually, your point of the relationship to the local translation and maybe some local computation sounds like a very reasonable assumption, but we don't know yet actually how they are linked to each other.
Sure, yeah. And could you also maybe comment on how, for instance, stimulating 7 spines or getting plasticity at 7 spines doesn't just cause a saturation of these signals very quickly and all the time? I mean, I guess that implies that
yeah, so that's actually also a great point, because such a few spines can activate nucleus to almost saturating signal. It gives almost saturating signal. We actually don't see much after 7, even 3 or 4. So this is very strange because, as you say, probably they receive LTP all the time, and if they can integrate over 40 minutes. So it's certainly they can be saturated. So only thing I can imagine is actually there must be some signal to reset or decrease. Unfortunately, we didn't find any of this signal yet, but that'll be very interesting. Actually, what's the signal to reset the status of the nucleus?
Thank you.
Okay, other questions for Ryohei? No. Okay. Thanks very much, Ryohei. Awesome talk. And Yueqing, awesome talk as well. Good to see everyone. Welcome back.
Thanks, Ryohei. Thanks, everyone. See you all next week.
Bye, everyone.
Bye.